Hello, everybody. This is the scientific method lecture, so I hope you enjoy it. So, when we think about science, we should think about how we do science, and that's what this lecture is kind of all about. Now, there's two general ways in which science is undertaken. The first form is just discovery science. Now, this is, basically going out and finding something new. So, we can think of that as, looking through a telescope and seeing something new out there. And every time we look in a telescope, every time we point the Hubble telescope in a new spot, we could potentially see something new that we've never seen before. The thing is, when you do discovery science, you need to take verifiable measurements, observations. So, writing things down, taking pictures, all sorts of things. So, some of the biggest ways that this has been done is by DNA was partly discovered in this way. They took a picture and saw that there was this double helix. Double spiral kind of shape, and found out that about DNA. The first biological cells that make up, organisms were found this way. New species. So oil companies were looking at these deep ocean vents, and they sent some robots down there, and they eventually found this whole new different type of ecosystem. So there's lots of ways in which this discovery science can happen, but realistically, most of the science is really done as hypothesis-driven science, and this is looking at following the scientific method. Now, the scientific method is your book might have something slightly different, but it's generally you make an observation, you ask a question about that observation, you make a hypothesis or a prediction about that, you test it, and then you get an answer. And then you should repeat it to see if you get the same answer. Now, real science often can, deviate from this method. And what I mean by that is people might jump in at different parts of here, where they might just jump in at the repeat. If someone else does an answer to a scientific question, another research group might be like, well, let's see if we get the same results so we can help them out. Someone might say, already have made a hypothesis or a prediction for you, and then you just kind of jump in on the test kind of thing. I think this can kind of be the how the scientific method works can be a little tricky to think about. So what I wanted to do is just look at one of the experiments that I did in the past. So I used to live in Kansas, and there's the Kansas River going through there. And this was part of my graduate work, and I think it's kind of looking at the whole scientific method, the whole scientific process in a relatively easier way. And I think it's you get to know a little bit about me and what I'm interested in, and then you also can see the whole process from start to finish in a real world example, not some little really simplified thing in a textbook. So I made an observation that the Kansas River, it flows through most of Kansas, can have these super big floods. So this picture was taken, and this picture was taken 3 days later. So what you can see is there is a whole lot more water, right? Look at the pylons in these bridges. There's actually about 26 more feet of water here in the river at this point than this point. And the cool thing about it was about 2 weeks later, the river was down to this level again. So, I'm an invertebrate biologist. I look at basically water bugs. So my question was, how do the bugs, so these are some of the bugs, these are basically like little mosquitoes, but they don't bite, called midges. how do the invertebrates in this river, the Kaw is that river, cope with these floods? And I had that hypothesis that sandbars in the river would make these refuges for the things. Okay, so that was my hypothesis, and I made this more specific prediction. I really don't care if you know the difference between a hypothesis and a prediction. They're pretty much interchangeable, at least if when you're talking to non-scientists. So my prediction basically was the places where you have more sandbars, or the times when you have more sandbars, the more bugs you would find in the river. So this is the same spot. This is at high water, and this is at low water, and you can see the river is very, very different. So what did I do? I basically did a field experiment where I went out and got to drive this cool boat and sampled a bunch of bugs in the river. And there's different ways to do experiments. You can just basically observe things. You can manipulate things. This was a whole big river. This river is about 200 meters wide. There was no way I was going to be able to manipulate or change anything about the river. So it was just a field experiment. You could do some modeling also then. So what did I basically went to the river, sampled the bugs in a bunch of different spots, and saw what happened. And basically my results were, where there were sandbars, there were more diverse, more species of these bugs. When the river had all these sandbars, more complex river, you had this more diverse time. So, and they actually use these sandbars as refuge areas. Basically, they would recolonize around those sandbars first before they would recolonize the rest of the river. And you can see that all this is a fancy graph showing that when the river was high and not complex, there was basically no sandbars. There was a different community of organisms. So one thing I wanted to do was then make some recommendations about why does this matter, right? Why do we care about sandbars? Why do we care about bugs? And we can get to some more complicated questions then that maybe other scientists can look at. And hopefully the conclusions that I made could shape the decisions. I mean, I'm sure there was some bias in my experiment, but I was hoping that my experiment could shape the decisions about managing the Kansas River and how they develop it. So, and that looked at this. We can see that by looking at this graph. Now, what this is looking at the river from upstream in kind of like the middle of Kansas, and then as we go to the east towards Kansas City. And you can see there's a big city here, there's another big city of Lawrence, and a big city of Topeka. And basically what they do is they have levees that are big dikes along the river that hold the river in, and so when it floods, it channelizes the river to narrower, and when it floods, it doesn't. When there gets to be more water, it doesn't flood, I should say. So that even though there's 26 feet of water in that river during that flood that I was showed you, there really isn't floods. But the thing that happens then is the complexity of the river. So the y-axis on this graph is basically how complex the river it is, how many sandbars. So the higher you are on the graph, the more sandbars there are. And what we see is around these cities where they have these levees, you don't have very many. Actually, if this graph gets to 1, you would have no sandbars whatsoever. And we can see it's almost approaching that, where around the cities where these levees are, you don't have any sandbars. So who the heck cares, right? They're these little bugs in the river. They're not even mosquitoes or anything that would hurt anybody. And that's probably true. Other than me that cares and likes to look at these bugs, nobody cares about them. But a lot of people care about these things in the river, right? When you go in a river and you see a bunch of people fishing, they're hoping to catch these fish. And in the Kansas River, you can regularly catch 60, 70 pound catfish, gar that are 40 inches long or so. And what is the food source of these things? Really, if you look at what these things are eating, especially this blue catfish, the base of their food web, are these bugs. Without these bugs, you do not have these big fish. So when you go back to think about this graph, as we get rid of these levees, we should expect that those big fish, as we add these levees and as these towns get bigger, the sandbars will go away, those bugs will go away, and then those big fish will go away. So that's kind of like from start to finish looking at my study. But my study is somewhat limited, and they're just like in all studies, whatever type of scientific study you're looking at, you need to really think about the scope of the question and how the samples were taken. And this kind of extrapolation example is, I think, pretty funny, where you can see that number of husbands that this woman has from yesterday. Obviously, today she got married. And okay, so we have 2 data points from yesterday and today. Tomorrow she should have 2 husbands. The next day, 3 husbands, right? As you can see, by late next month, you'll have over 4 dozen husbands. Okay, so that's what extrapolation is. It's going beyond your data, given the trend that you see of where you sampled, of where we've only sampled yesterday and today in this example. So when we look at my study, so what does my study on this Sandbed Prairie River in Kansas say about the Amazon River in the Amazon rainforest? Well, pretty much nothing, right? The Amazon River reacts very differently. It's way, way, way bigger of a river. So my study can't really say anything about the Amazon River. But what about other sandbed rivers? What about a river that's next door in just to the north in Nebraska? From Kansas, next state up north is Nebraska, right? You might be able to make some qualifying statements that say, since we found result A in the Kansas River, potentially that is in the Platte River in Nebraska, somewhat similar. But so science should be limited by the scope of the question and not have too much extrapolation. The thing is, political groups or people that have a conflict of interest might grab onto this as proof. And we can see this really happened back in the 60s through kind of the 90s, where the cigarette companies were really trying to defend their product and saying, well, cigarettes really aren't addictive or bad for you. Scientists basically did a bunch of studies looking at, let's say, nicotine. And scientists found out that nicotine causes disease, right? It causes cancer. It is itself a carcinogen. But then the tobacco companies were saying statements like this: scientists have not proven that cigarette smoke or any of the thousands of its constituents as found in cigarette smoke, and that's key to this statement, as found in cigarette smoke cause human disease. So they were saying, well, you just looked at nicotine. You didn't look at nicotine in cigarette smoke. And, we obviously know that to be completely false. There is ways to do science, look at specific chemicals, or in this example, that you can do a little extrapolation and go a little bit beyond your original question to show certain things. So let's go back to my my graduate work in Kansas. And what I was doing, I wrote 2 journal articles, one in the Journal of the North American Benthological Society entitled Flora Refugia for the Zoobenthos of a Sandbed River and the Role of Habitat Complexity. Actually, this is a different project here, but again, this is from some of my graduate work from the Journal of Freshwater Biology. And I'm sure all of you have read that, right? You've all read the Journal of the North American Benthological Society. Really what we see is there's really a disconnect of how we do science and then how we present it to the public. There are some like popular science magazines like National Geographic, Scientific American, things that you might have seen before. And the thing is, these articles or these publications do a good job of highlighting like specific science, but it's really just a very small sliver of all the science, the important science that's done all around the world. And they usually, think about the real sexy ideas, not necessarily what happens to bugs in a river when it floods, right? And I think that's okay, but there is this huge disconnect between, the people actually doing the science and then getting it to the public. I think some of the things that help with that, though, which is certain websites like IFL Science. It's this the kind of like the lighter, they call one of their subtitles to their website is the lighter side of science. It started out by a graduate student, and she was just really upset with how science was projected, and she created this website, I Fucking Love Science, and there's I think this was in the mid 2000s, but it's exploded and gotten a lot bigger now. I don't really love the way the articles are written now in the last maybe 5 years, but it still brings up a lot of really cool studies and presents scientific ideas in kind of a different light. Another really famous scientist, astrophysicist named Neil deGrasse Tyson, he's got a pretty cool Twitter account where sometimes he live tweets like sci-fi movies, and as he's watching them to complain about the bad science. Basically, he does it in a funny way, but he has a lot of really cool tweets that are science related and highlighting the importance of science in our world. Another one that we will actually become really familiar with in this class is Radiolab. It's a podcast out of WNYC in New York. It's a pretty cool podcast all about different types of science, neurobiology, ecology, almost all types of science. And they do a really good job of really doing these in-depth stories and making a really cool a really cool podcast. We'll be using a lot of them as our podcasts and our podcast quizzes. One other thing I want to talk about the process of doing science is there's a lot of really bias in publishing, and we see the huge bias towards significant results. Okay, and if you might think about this, is that scientific careers are not made on negative results. So think about my study that I did in the Kansas River, the bugs, basically needing these habitats, needing these sandbars to survive. What if I had found the opposite of that these bugs don't need the sandbars? These bugs do just fine, and they don't need to, these sandbars are unimportant. Is that isn't interesting of a story? In some ways, okay, it's kind of interesting, but it's really a less interesting thing. What that means then is if I had found that other thing, if I had found it the other way, it probably would have meant that journal article might not have gotten published. And that might not have been that big of a deal because it's just river bugs somewhere in some river, right? But in things like biomedicine are extremely important. Finding out if a certain drug does or doesn't work, and let's say you find out it doesn't work to cure some specific disease, that's really important. So, a really current example is with the coronavirus pandemic, where there's been a lot of press coverage on this drug called hydroxychloroquine, where it was this anti-malarial drug that was hypothesized to have some good benefits on it might have helped as an antiviral drug. And it turns out it wasn't effective. We see that there isn't really a benefit to using this drug. And that's super important so that we don't waste any more time trying to use this hydroxychloroquine drug when we know it really doesn't work. And, negative results at times can save lives, can really help progress science. All right, so that's all I have for the process of science. Hope you enjoy it, and see y'all later.